Just listening to you guys, I mean, obviously you've done a lot of research in the field of omics. Steve, when did Purina start doing genetic and genomic research? How did Purina get into the field, and then you guys can talk about sort of where it's come from there.
I think what Rondo just said about how far we have come and how far we still need to go fits that question quite well. When we were looking at osteoarthritis on the transcript level, the pool of RNA, the genes that were up and down regulated, you mentioned about 2,500 candidate changes that we saw. When we went to find out what proteins, because the RNA is just a code, it's just the instructions, what proteins did those 2,500 RNAs code for so that we could try and figure out what biology was going on, I think it was less than 20% had been annotated, meaning we didn't know what vast majority of those mRNAs that were changing in arthritis, what were they coding for? And so it was clear that we needed geneticists to tell us what the code of the genome was. At the time, the human genome was being sequenced, but not a lot in canine and feline. So Purina started engaging with geneticists who were working in the canine and feline area. Concurrently, there was a group from the Canine Health Foundation, which was a research foundation from the American Kennel Club, was in looking at the similar type of efforts. And so we combined forces and were able to bring together a global group of geneticists who were often coming from the human genetic world, and interestingly, now almost all of them are exclusively canine and feline geneticists because they just fell in love with pets as we all have. But nonetheless, we were able to bring those together, those scientists together, understand what their limitations were. For example, they were all placing markers on the DNA because they weren't sequencing the canine and feline genome then. They were just simply placing landmarks along the DNA.
What exactly do you mean by placing landmarks? I know they're identifying portions of the gene, but
right. So they're looking at pieces of DNA that are different. Okay. Located in the same spot but different between animals, and so genes that are close to those markers will move together, and it's called linkage. But nonetheless, these scientists were all placing markers on families of animals around the world, and what was needed was to place those markers on a common reference family. And we were able to work closely with Cornell University to identify a family that they had, collect that DNA, we housed it here at Purina, and then had a distribution program to send that out to all of the geneticists so they could place their markers on a common family of DNA, which was huge in allowing us to map at least proximity of where genes were. And maybe more importantly, in the long term, as the human sequencing was completed and these sequence centers were looking for next species to sequence, the canine, and then not far behind it, the feline, were rapidly elevated in priority, much due to this wonderful group of geneticists who had, I think we facilitated a lot of their ability to prioritize the canine and feline genome for sequencing, which has been done now, of course.
Okay, good. I want to follow up on that, Rhonda. You've been involved with some recent enhancement on the feline genome?
Yeah, on the feline side, yeah.
Briefly share with us what was, I mean, because it led to an interesting publication, so what was special about it?
Yeah, we've had a few publications. So we, as Steve mentioned about putting markers on a map, it's really like a road map, so you have a reference to know where you are studying on the chromosome or the DNA relative to those markers. So I think there's really two areas. Steve briefly mentioned linkage. So there's a linkage map, a linkage reference genome. And it's It's not based on absolute distance. It's based on, try not to get too technical here. It's based on how often those areas in the genes recombine during meiosis.
Okay.
So I think that's a little technical, but different regions on the DNA recombine and at different rates. And so you have to place markers
Spaced correctly,
basically. But the work that we were able to do with Mizzou,
University of Missouri.
University of Missouri, as well as Texas NIH, Texas A&M, UC Davis was involved in that as well. We were able to place over 55,000 markers on there, and it increased the magnitude of markers greater than 20-fold. So it was a vast improvement.
So there's this mapping technology that Purina has been a part of through collaboration with leading scientists probably all over the world, right?
Yes.
You mentioned some U.S. universities, but I think you have collaborators around the world working in this
Sure.
area. But so you have this mapping function, very important. How does that tie into the omics studies? Are they totally different? Do they overlap? Does one feed into the other? How do they play with each other?
Rondo, I'll let you follow up with that. I guess what I would reiterate is our initial push into the area of genetics and sequencing was so that we would understand what the code was. So when we saw a change in a particular RNA, we would know what protein was going up or down. So that's the link back to nutrition. What are the switches? How is it up and down regulated, as we talked about before? But what we're finding now is that the whole area of genomics and understanding the potential an animal has, the risks an animal has, the opportunity for them to be faster, stronger, smarter, all of that is giving us new directions for the company to move, and we're moving in those directions. So understanding the genomes more deeply, as Rondo and team have been doing, just gives us more information about dogs and cats and how we as a company who aspires to do best for dogs and cats can utilize that information.